You see it all the time, right? A headline pops up about some big medical breakthrough, and the article says the results are statistically significant. And that just sounds so solid, like it's settled science. But what if I told you that powerful little phrase could be hiding a much, much weaker story?
So let's get into it. How do we actually know if a study is the real deal? When scientists are testing a new drug, they're basically trying to find a clear signal in a whole lot of noise. They have to be sure that the good results they're seeing aren't just a total fluke, like a lucky roll of the dice.
And for, over half a century, pretty much the entire scientific world, we're talking researchers, doctors, even journalists, they've all relied on one single gatekeeper to make that call. One number that has decided which treatments get the green light and which ones we're told to trust with our health.
And here it is, the famous p-value. To be specific, a p-value of 0.05 or less. This has been the absolute gold standard, the magic number that screams, hey, this probably isn't just random chance at play. But, as we're about to find out, that number, well, it doesn't even come close to telling the whole story.
This brings us right to the heart of the matter, the significance problem. Because it turns out relying on just the p-value to decide if a study is strong or not, it can be seriously misleading.
The best way to think about it is like this. A p-value is kind of like a black and white photo of the evidence. I mean, it's useful, for sure. It tells you if something is there. But what it doesn't do is give you any of the color, the depth, or the details. You can't tell how sharp or stable that image really is.
And this, right here, this is what blows the whole thing wide open. Look at this. Both of these trials are officially statistically significant. They both have the same p-value of 0.04. But trial A? It's pretty solid. You'd have to change the results for 10 different patients to make it not significant. And trial B? It is hanging on by a thread. Just 2 little changes and the whole conclusion flips. Yet the p-value makes them look like identical twins.
So you're left with these 2 massive unanswered questions. 1, how stable is this result really? Could it just fall apart with a tiny change? And 2, how strong is the effect? Like, is it a massive improvement, or is it just barely better than doing nothing at all? The p-value just can't tell us. Okay, so that's the problem. But now for the solution. A newer framework is here to give us that richer, kind of 3D view of the evidence. And it does that by introducing 2 really cool new concepts, fragility and robustness.
It's called the evidence triplet. It's super smart. It doesn't throw out the p-value. That's still there to check for significance. But it adds 2 extra layers. First, fragility, which tells us all about the result's stability, and second, robustness, which measures the actual strength of the effect. Let's break these 2 down.
All right, fragility sounds complicated, but it's actually a simple and brilliant idea. It just asks one question. What's the absolute minimum number of patients whose outcomes would need to change, say someone who recovered now didn't or vice versa, to flip the study's result from significant to not significant? If that number is low, the study is fragile. If the number is high, it's stable. Simple as that.
And then there's robustness. This one is all about signal strength. It puts the result on a simple scale from 0 to 1 and tells you how far away it is from the no effect line. So a really high robustness score means you've got strong, clear evidence that the treatment is actually doing something meaningful.
So, okay, this all sounds great in theory, but what happens when you apply this new framework to real-world studies? Well, a recent study did exactly that. They took a whole bunch of clinical trials and put them to the test.
The researchers grabbed 77 different clinical trials, and remember, all 77 of these had already been published and declared statistically significant based on that old p-value standard. Then they put every single one of them under this new, much more powerful microscope.
And what they found is kind of stunning. It turns out that the significant label was masking a huge difference in quality. Only about 39% of these trials had what you'd call truly solid evidence. But get this. A massive 31% were what the researchers call significant, fragile, weak. That means, yeah, they passed the p-value test, but they were also incredibly unstable and had a really weak effect.
So let's make this crystal clear. The Concordant-Positive results, or CP, those are the gold standard. They hit all 3 marks: significant, stable, and strong. You can feel good about those. But the significant, fragile, weak, the SFW results, those are the danger zone. They got the official stamp of approval, but they're really built on shaky ground.
Now here's the part that really proves this whole framework is onto something big. The study found this top-tier, solid evidence in 39% of the real published trials. But then, they ran computer simulations where they knew for a fact there was no real effect, it was all just random noise. And you know how often that solid evidence pattern showed up by pure chance? Only 1.4% of the time. That's incredible. It means real science is finding solid evidence nearly 28 times more often than randomness ever could.
And this really is the bottom line. The study's authors give a pretty stark warning. They say that these SFW results, the ones that are technically significant but are also fragile and weak, they're the ones that absolutely demand caution. They need to be double-checked, replicated, and looked at a whole lot closer before we start betting people's health on them.
Look, this isn't just some nerdy statistical debate. Getting a clearer, more honest picture of scientific evidence has massive real-world consequences for the future of science, sure, but also for our own personal health decisions.
So if there's one thing to take away from all this, it's this. Not all significant findings are created equal. That single label we've trusted for so long just isn't enough. Behind it, some discoveries are rock solid, but a huge chunk of them, nearly a third in this analysis, are just hanging by a thread. By adding these new tools, fragility and robustness, to our toolkit, we can finally see the difference. We can sort the truly convincing results from the wobbly ones. And that's going to help improve the quality of all science, and most importantly, it's going to help doctors and all of us make much better decisions based on much stronger evidence.
So I'll leave you with this. The next time you see a headline or read a story about a significant new finding, maybe your first question won't be, what's the p-value? Maybe now you'll ask, okay, but is it stable and is it robust? This entire breakdown was based on the fantastic research from a study by Thomas F. Heaston. It was published in the journal Curious, and it really does lay out a powerful new way for all of us to think about scientific evidence.